Okay, I think we can go ahead and get started. So, hello everyone. Thank you for joining us today. My name is Mallory Grunner, and I am Head of Events at BioMed X. I will be moderating today's webinar on the open call for application from BioMed X and Servier, a new AI-empowered platform for sterically guided design of biospecific antibodies. Today's speakers are Thomas Rucke and John Linde from BioMed X. And then we also have Véronique Blanc and Clément Barjon. And we also have Celine Truquel from Servier. And Celine will join us later on in the webinar. So they each have a short presentation prepared, and then we will have a time for a Q&A session afterwards. But before we get started with the presentations, we'll just have a quick round of introductions. So Thomas, would you like to start us off?
Yes, Mallory, thank you very much. Yeah, hello everyone. Good morning, good afternoon, wherever you are in the world. My name is Thomas Rückle. I'm the head of research here based in Heidelberg, and I oversee all the projects of European projects of BioMed X.
Thank you, Thomas. John, would you like to go next?
Good morning, everyone. My name is John Lindner. I've been with BioMed X since 2018, currently in the role of head of research for the US-based projects, and in today's call in my role as head coach of the bootcamp and recruiting processes.
Thank you. Véronique, would you like to go?
Yes, hello everybody. Very happy to be there with you all. I'm Véronique Blanc, and I'm leading the immuno-oncology research group at Servier.
Thank you. And last but not least, Clément.
Yes, hello everyone. I'm Clément Barjon, and I'm a research project leader in immuno-oncology at Servier. And happy to be here also.
Perfect. Thank you very much. So before we get into the talks, I will just explain a little bit of the housekeeping for the webinar. So if you haven't used Zoom webinar before, please look for a box that says Q&A. We kindly ask that you submit all of your questions via this Q&A box and not via the chat box. And you're of course welcome to submit all of your questions throughout the entire presentation. And then once we finish, we'll get to as many questions as we can during the Q&A portion of the webinar. As you can see, this webinar is being recorded, so it will be available in 24 hours on our YouTube page. And I will be posting a few links in the chat, including the link to apply for this call, as well as the link to our BioMed X career space. And if you sign up for that, then you receive updates about this call for application, as well as information on future calls. So without further ado, we'll start off with a presentation by Thomas and John, and they will share about BioMed X, the application process, and the bootcamp, which will be in Paris-Saclay. So I will share my screen.
Thanks, Mallory.
Okay, can you see my screen?
Yes, perfect.
Okay, perfect. Then the stage is yours, Thomas.
Thank you very much. So yeah, as Mallory already introduced the topic of this call for application today, new AI-empowered platform for sterically guided design of biospecific antibodies, in short ADP, in partnership with Servier. You may wonder what BioMed X is all about, and so I'm going to give you a little overview about BioMed X, because this could be a potential future employer. Can you go to the next slide, please, Mallory? Okay, yeah, BioMed X Institute is an independent research institute that's located in Heidelberg. It started actually about 12 years ago on the campus of the University of Heidelberg. It's now expanding, and it's embedded in the campuses of the University of Heidelberg, as well as a US side, closely related to the Yale University. And what's really unique about the BioMed X Institute is that we're combining the best of both worlds, academia on one side and industry on the other side. And we do this through a unique innovation process that was published in Nature Biotechnology in 2015. Going to show in more detail what this all is about. So we seed biomedical innovation by combining the best of both worlds, as I just mentioned. One world, the industry world. What do we get from the industry? We get what we call challenges, projects, tough nuts to crack, projects that would really make impact for the pharmaceutical partner down the line, 5, 6, 7 years. The industry partner also provides mentorship and obviously the funding. On the other side, the academia, where we would get the talents from, you guys who are on the call today, the project ideas, mentorship as well from professors at universities. We would have access to human samples, academic IP, and obviously the infrastructure on the campuses that we are here in Heidelberg as well as in Yale. And all that together then would allow us to provide solutions to the industry, in this case, Servier. This could be drug targets, could be biomarkers, could be platform technologies, or what's very pertinent for this call here, AI models to solve tough questions. So I mentioned earlier that we have a process in place. We built from scratch what pharma needs next, and we call it innovation as a service. This first part is already the past to say that we actually hold workshops with pharmaceutical companies where we identify top challenges. We also identify internal champions for a call, for a project, and we explore, articulate a couple of challenges and identify one, which we then would articulate, as you can see now on our crowdsourcing platform. And John is going to introduce a bit more about that. A crowdsourcing platform that allows you to apply not only with an application letter, but with a fully fledged project proposal. And John will explain in more detail what this is all about. We then would select out of 200 to 300 applicants who apply with a full application, the 15 top talents and project ideas, obviously. And we would bring those people then for 5 days to a boot camp in which we would coach for 5 days, 5 different teams in certain directions, such that the pharmaceutical company after those 5 days has truly choice to say, this is a really interesting project. We would like to fund that. Let's go. And then starts the co-creation, which is then obviously the exciting part also for any of you to be part of a BioMed X family, to be part of a 3 to 5 years project, building innovative solutions in IP. Obviously, this is not done in isolation. There will be mentorship and there will be a lot of guidance from all different parties. This is a photo of a recent bootcamp that we had in collaboration with our second Japanese pharmaceutical company, Daiichi Sankyo. This was a bootcamp about strategies for targeting solid tumors with multi-specific biologics. So new ideas around that topic. This was held in Heidelberg, Germany. So BioMed X, as I mentioned earlier, has been on the block for the last 12 years, started in 2013. And you see here a list of 12 pharmaceutical companies that trusted us so far in collaboration. Here in Heidelberg, where everything started, we initiated up to now 24 projects in the field of oncology, immunology, neuroscience, as well as one project in artificial intelligence. Recently, 2 years ago, we expanded to the United States, and we have a first project there, as I mentioned, close to the campus of Yale University, very prestigious university. Next, we then actually expanded our model, not on a territory level, but more from a philosophical thinking. We started in 24 the so-called XSeed labs project with Boehringer Ingelheim, again in the United States, in Richfield, Connecticut, a project in the field of immunology. I will come to this in more detail in a second because it's also pertinent for this call. And what's also pertinent for this call is that we have expertise in setting up startups in the field of artificial intelligence. Together with Ion Labs, we have so far generated the creation of 5 startups in the field of artificial intelligence, and this in Israel. And lately, in 2025, we won a pitch at the Gates Foundation where we will set up startup companies in the field of women's health, and we will soon start our first call in the so-called XFAM Labs environment. Yes, I mentioned. So can you go back? Sorry. So I mentioned, yes, the XSeed Labs is very pertinent to this call because it's a different model to the normal classical biomedics projects, which are at the sites of biomedics. The XSeed Labs is actually a biomedics project in a box at a pharmaceutical company. Next, please. Yeah. So XSeed Labs started, as I mentioned, in Ridgefield, Connecticut, in collaboration with Boehringer Ingelheim. The teams there are embedded with the pharmaceutical company's ecosystem. That's very different to the projects that are here in Heidelberg or at Yale University. These teams have access to local expertise and to the infrastructure, and in close networking and collaboration with the R&D colleagues on site. And the same is here for this call. For Servier, it's going to be an XSeed Labs at Paris-Saclay. So Paris-Saclay, where is Paris-Saclay? Yeah, this is a photo from back 2019, how the lab's Servier site looked like. And you may wonder, how can I do research here? But there is 2019 written. And this is how it looks like 6 years later. The XSeed Labs Servier site, it's actually the Servier research site at Paris-Saclay, a fully fledged research building, a fantastic building with all possibilities. And there's also the Spartners, which is a project that is going to be introduced by Celine in a second, with an employee of Servier. It's the right part of the building here at Servier. And this is a fantastic facility, a fantastic opportunity for you to join. Next. Yeah, so what's in for you? Fully funded 3 to 5 years research, focus on outstanding research and publications. So you work towards very targeted goals to help the pharmaceutical company move the needle in their needs. But you have the opportunity to publish so that you don't have a gap in your academic career in case you decided to go back to academia. A creative cross-cultural teamwork atmosphere. At the moment, we're about 60 employees here in Heidelberg, and every other person has a different passport. That makes it very exciting from a cultural perspective as well. You will be guided by experienced mentors from academia as well as from industry. And in this particular case, because you are on site, the mentoring will be very, very intense because it's very close. Outstanding environment with direct access to leading research institutions and companies. Anything you cannot find in your own lab, we will make it happen through a network that we have established. You will get leadership training, you will get entrepreneur training, you will get the right coaching that in these times at biomedics, you also see a progress, an upgo in your personal career. And it's a risk-free assessment of possible future career. So you may want to go to a startup, you may want to go to a big company, you may say, no, thank you, I want to stay in academia, I publish my papers. It's an opportunity for you to explore in the risk-free environment what you're really up for. The hiring process, yes, one more slide, one more click, please. Yes, so yeah, as I mentioned earlier, we're going to have this exciting boot camp of the 15 most brighted talents from you guys applying. Out of this boot camp, there will be winners, group leader, postdocs, 2 postdocs, 2 research assistants, who will receive an employment contract offer from BioMed X, but physically they will be at Paris-Saclay in the Spartners building next to Servier. We will help you relocate to Paris-Saclay or Paris, work visa, finding home, moving your family, kindergartens, and the whole spiel. And the anticipated start date for this call and this project is early February 2026 in Paris-Saclay. And with this, I'm done with my part about biomedics. If you have any questions, then please post them as instructed by Mallory. And I'm handing over to my colleague, John. Please, John.
Okay, great. Thank you, Thomas, and welcome also from my side. And perhaps by now you're excited about the prospects that BioMed X has to offer, and you're wondering, am I a BioMed X type of person? What does a BioMed X fellow look like? And I would say globally, no matter where we are in the world or where we come from, what unites us are our 3 core values. And I'm going to propose that these core values in recent years have become somewhat controversial, and I'll explain why. The first one is to cross-leverage. We inspire and motivate each other to unleash our full potential. Why might this be controversial? Very frequently, we come across hiring managers that look for a specific profile, exactly the right expertise for exactly the right position. I tend to reject this notion. I find that at BioMed X, the individuals that we look to hire are the most critical thinkers, the most creative problem solvers. Of course, you cannot be a plant biologist and apply for an AI call in bispecific antibodies. This would be a bit too out of scope. But don't necessarily consider yourself out of the running for a position like this. We really, and you'll hear about this from Clement in a minute, have an idea of a problem, but not a solution. And the proposals for those solutions can come from anywhere. Once you join BioMed X, cross-leverage means that you join our global community of scientists who have orthogonal scientific backgrounds with respect to protocols and approaches to problem solving. And so you will get to interact with those even from XSeed labs via a virtual global seminar, virtual meet and greets, and from Paris to Heidelberg is not that far. So you will get to join social activities to interact with your colleagues at the BioMed X main site. The second core value is to cross-pollinate. We embrace diversity as it is the source of innovation. We are also a US-based company, and given recent changes to the politics there, this has become a very controversial statement to make. We stand by it. We believe not only in terms of scientific background, but also in terms of cultural background, creating a diverse work environment is key to success, especially in high-pressure, short-term projects such as these. It's important to bring together a team that works together both at the interpersonal level as well as the scientific level. And we have seen this with all of our teams, that having diversity enriches and synergizes the project rather than detract from it. And finally, aim high. This has become controversial in my opinion because I was recently told it makes more sense to underpromise and overdeliver if you want to impress the higher ups. This is a notion that I cannot get on board with. At BioMed X, we never underpromise. The goal of the boot camp is actually to come up with a sky-high proposal that will change the world and change the way that drugs are made. It's then the job of the winner to figure out how to make that proposal a reality, and you have 3 to 5 years to do so. Thomas and I will do our part to convince pharmaceutical partners that we're not overpromising, and have some understanding if we don't meet all major milestones, but make sure that there are deliverables that still move the needle. In my opinion, underpromising just creates a very dull, boring project. So be prepared to think creatively and aim high as you prepare project proposals. Those are our core values. Just to give you an example of how we cross-leverage within our organization, we can go through the animations very quickly here. At the moment, we have several focuses of our research groups between wet lab and dry lab, a lot of expertise in building complex in vitro models, pairing that together with our machine learning, artificial intelligence groups, or the dry lab group that we have in Heidelberg at the moment as a prime example of this. And then moving slightly away from the biology and into the biotechnology side, developing a lot of sensor technology, developing a lot of platform technology that enables and empowers these two other circles of the Venn diagram. And so these are all examples of projects that we have currently ongoing or have recently closed off within our 4 walls. So you've heard the term boot camp several times, and maybe wondering if you haven't heard about BioMed X before, what does this look like? So the boot camp for this call will be at the end of November in Paris-Saclay. And it's basically 5 days of fun. And you'll see what I mean by that. Day 1 is actually, I would say, very pleasant socially. Everyone gets to know each other. We have invited 15 individuals to participate, plus our entire team. This is coaches from CFDA. This is coaches from BioMed X. We will introduce ourselves. We will have a great time getting to know each other. And what's very important is you will get to know each other scientifically, because at the end of day 1, you will rank your preferences for whom you want to work with for the remainder of the week. We choose the teams at the end of the night and let you know at the beginning of day 2, which is when everyone gets to work. We split off the 15 individuals that are participating in the bootcamp into 5 teams, each led by a single group leader candidate, who are then instructed to further develop, potentially synergize, potentially revamp their scientific proposals based on the brief that you will receive from our sponsor on day 1. Your proposal may have been fantastic, but it may turn out not to have been completely in scope. And this is what the bootcamp is for. If it was a perfect proposal, we want to see you improve it. If it was slightly out of scope, but you seem like a very strong candidate and capable of some flexibility, we will provide you feedback with how you can improve your idea into a central hypothesis that fits the mission of the project. Day 2 is mostly about brainstorming and literature research. Day 3 then is the start of the work of turning that idea into a concept that you can pitch in a 15-minute slide deck to the jury on the final day. And this is really slide building day, where you take those ideas, that background literature, those proof of concepts, put them on paper, state the state of the art and the problem that you're going to solve, and give us your firework proposal for a solution. Day 4, we see for the first time how all of the 5 teams stand with respect to the proposals that they have developed over the previous 2 days. This is the dry run of the presentation you'll be giving on Friday. And you'll have the rest of day 4 to improve based on about 30 minutes worth of feedback you receive from the coaches and from your colleagues among the other attendees. Keep in mind, the whole time, while this is primarily about the science, the coaches are observing how the teams function, because we don't just hire based on the project proposal. We hire based on leadership skills. We hire based on response to feedback. We hire based on ability to work together in a team and become more than the sum of the parts. So this is very much an assessment that's happening as the bootcamp progresses, but it's not something that you should have in the front of your mind. Just be yourself, be a brilliant scientist, and you will stand out. Finally, day 5 is your opportunity to shine. The Joint Steering Committee members who aren't necessarily coaches during the bootcamp will attend and hear your pitch. And based on the presentation that's made on day 5, the decision will be made for who to follow up with and offer the positions within BioMed X. So that's it in a nutshell. 5 very crazy days, very little sleep, especially in the middle. And that, to be honest, is when some of the best ideas pop up. And we've seen it before, and we hope to see it again. You may see your current affiliation on this slide. This is an ever-growing list of where BioMed X sources its fellows from. And so you see we bring people in from across the globe. And while they're with us, they are also encouraged to collaborate and publish. So the next slide, you'll see many lists of the collaborations we have ongoing or have wrapped up. I mentioned publication. This is one of our, let's say, sub-core values underneath Aim High. We look to develop solutions for our pharmaceutical partner while simultaneously in the form of intellectual property, while at the same time offering our scientists a path forward in their career by finding ways to make sure that they can publish in peer-reviewed journals based on the work that they provide. And there's always a process for this together with the pharmaceutical partner. And it has always worked out. As a good example, you can look into my previous work. I joined BioMed X as a group leader and spent several years working on developing a platform that has now been published, and I'm very proud of that work. And simultaneously, it was onboarded within our pharmaceutical sponsor. Now, projects are not eternal at BioMed X. We do look for turnover to continuously bring in fresh perspectives and ideas. So when our fellows leave, where did they go? I would say we have a very good track record of sending individuals both toward academia and toward industry. So it really is the best of both worlds, especially for young scientists who still want to work at the bench, but are eyeing a future direction in either industrial scientific work, positions outside of bench science, or returning, or if they've been close to it with the project, staying on that academic track. So what do you do next? First, you listen to the remainder of our webinar. You're going to hear about the details of this particular call. If you haven't already, you go to our website. and register your interest in the call. Now, the call itself will require you to upload your profile, including your CV and a motivation letter, and most importantly, a short project proposal addressing the challenge. You're going to hear the challenge in a little bit more detail in a few minutes. My suggestion to everyone submitting a project proposal is to make sure you understand the scope of the project. Your proposal can be short as 2 pages, and we are looking for creative sparks. We are not looking for proposals that reinvent the wheel. We are not looking for proposals that merely state the problem in their own words. We are looking for something to that proposal, maybe not in entirety of detail, which you'll work out during the bootcamp, but something novel, and make sure that it's within the scope of the problem you're going to hear. Send that all to us by the end of August, and it's not very much luck-based, but it's very much skill-based. You will be hearing from us within the following 2 weeks, potentially with an invitation to join us at Paris-Saclay in November for the bootcamp. And finally, in case we haven't mentioned it enough, we have a career space portal. If this call isn't for you, we do have calls that are published roughly once every 3 months. And by registering for our career space, you will be notified and you will receive our newsletter anytime there's a new call for a team or an open position in one of the existing teams. We'd love to have you join our family.
Perfect. Thank you so much, Thomas and John. So I see now Celine has joined us, so thank you very much for joining us. We're looking forward to hearing more about Spartners and about yourself. So I would just like to remind everyone that you can put your questions in through the Q&A box throughout the presentation, and then we will get to them after this presentation. So then we can move forward from hearing from Veronique, Clément, and Celine with their presentation about Servier, the Spartners location, and about the call topic specifically.
Yeah. Hello, everybody. I will start, and we will first start with presenting ourselves, Clément, Céline, and myself, so that you have a bit of idea of our background. So Clément on the first slide. I am Véronique Blanc. I have been trained as a microbiologist and molecular biologist a long time ago, and then I was trained for immunology. And I must say that I spent all my career in drug discovery, in pharma, in Sanofi first, and since 8 years in Servier, where I am leading the immuno-oncology research group. Clément?
Yes,
Oh,
so yeah.
Yeah.
So I'm Clément Barjon. So as I said, I'm a project leader in immuno-oncology at Servier. So I did my PhD a bit over 10 years ago in the Institut Gustave Roussy in Villejuif, near Paris. And so I did most of my career in immunology and more specifically in immuno-oncology. So I did 7 years in academic labs, across postdocs, and I did a bit, 3 years in Sanofi. And I just joined Servier 1 year ago. Yeah.
Céline?
Hello, everyone. So, yes, I'm Céline Triquel. I'm a pharmacist by training from Toulouse University. I'm now leading the head of the open innovation at Servier, for which Spartners is one of the main topics who is occupying myself a lot, and I will explain after. I have a long career at Servier. After this position, I had led a team dedicated to medical data and medical coding in the clinical development team. And then I was heading the lab Saclay, which was a lab of innovation before coming at Saclay. So I was pushed into the fabulous world of innovation from, I would say, 2018. And it's now a real passion for me. So I'm very happy to say more about Spartners just after the quick presentation of Veronique about Servier.
Thank you. So on the next slide about Servier. So I think the first time we would like to share is that we are governed by a non-profit foundation, and that is important for us as it means we have no stakeholders. And so as a consequence, we believe we have more freedom to invest long-term to develop a therapeutic solution to patients in a sustainable way. On the next slide, a few words about Servier. We are a mid-sized pharma company, second largest pharmaceutical company in France. Servier has been a leader in cardiology, since long, and first in hypertension. We are about 20,000 employees, and about 10% of them are involved in R&D. There is more than 30 drugs that are commercialized, mostly in cardiovascular, but a growing number in oncology and a few in neuroscience. Of the 6 billion revenue that we, for example, had in 2023, 2024, we spend about 20% in R&D each year, which means a lot for us and for our research. On the next slide. Yes, I wanted to say that in Servier, you will find a lot of experts in drug discovery, of course. But in fact, what I really liked is that you will find a lot of passionate people who contribute to Servier individual and collective engagement for patients. And also what is important on the next slide is to share that, of course, we are working for patients, but we are working with patients. That means that they are at the center of the medicine life cycle at all stages of our drug discovery and development. And this is very important as it allows us to respond better to their need. On the next slide. I want also to highlight the importance of our corporate and social responsibility commitment, among which for the employees, where we try to develop a safe and inclusive environment. And we have developed a number of diversity networks across all our sites. And we are also committed to our planet with different objectives like reduction of the greenhouse gas or maintenance of the biodiversity. On the next slide, a few numbers again. So here you have this pipeline that our commitment has allowed to build. So you have about 60 projects, 36 in development and 24 in research across oncology, neurology, cardiovascular, and we have a few legacy projects from immunoinflammation. On the next slide, and it's about our R&D organization that support our portfolio. We have 3 hubs, North and South America, Europe hub, and Asia and Pacific in 3rd hub. We have a lot of clinical development sites worldwide, a lot of production sites worldwide, and we have 4 research centers, 1 in US and 3 in Europe. And you will hear from Serine about the Saclay Paris Center, where we have a whole R&D activities. But of course, on the next slide, I wanted to highlight that we are not working alone. We have built a large network of external innovation, and whether we have partnership with biotech or pharma company, or on the next slide, we have collaboration with academics, among which you can see already BioMed X. And I cannot avoid to talk about our ShineDocs community on the next slide, which regroup all the PhD and postdoc, about 80 as of today, that creates this very active community, which connect very efficiently, and you would have a lot of potential to connect, discuss, and build together. On the next slide, I wanted to highlight one point before heading to Céline to talk about our R&D research center. I want to highlight that Servier is committed to invest a lot in artificial intelligence and data to support our R&D value chain. This is very important for us, not only for innovation, but also to improve our decision making and increase our efficiency. And we apply that along from target ID to registration. And obviously, we apply that to our molecules, small molecule and biologics, and the call is about that. Without going further, I leave you the floor, I think, Céline.
Yes, thank you. Thank you very much, Veronique. Yes, just to give you a little overview of what is our Research and Development Institute at Paris-Saclay and why we have chosen this super cluster, because it's where you would be embedded, in fact, and it's where you would handle this topic. So Servier has decided to merge all the R&D French teams in a unique site to enhance and increase the collaboration between each other, between the team, and to have a more efficient R&D. And we have chosen Paris-Saclay, which is the ACE-based cluster of innovation all over the world. So it's not trivial. We have around this building a lot of high schools, business schools, and the brightest schools in France, like Polytechnique, like Centrale Supélec, École Normale Supérieure, Paris-Saclay. So it brings a lot of brain and brightest students around us, and a lot of also public labs, INSERM, CNRS, which one we can collaborate. So it represents a very rich environment for innovation and a very stimulating environment for innovation. That's why we have chosen this cluster. So we have the chance to have a super building, which is it's a view from the sail, but it's a real view. We have, in fact, in the ring, in the wing, we have the labs. We have more than 3 kilometers of labs, which means a lot. So all our platform of R&D are there, and the research is coming from here. Also in Boston for the target identification in oncology. In Budapest, we have also a chemistry team, a very brilliant chemistry team on top of what we have here in Saclay. And in Barup, for sure, all the anticore organization with the former Symphogen company that we have acquired. So on top of this super building, where more than 1,000 people are working on R&D here, on the upper left side, you see a little building, which is an independent, in fact, building. It's a weird situation because it's not really Servier, but it's completely embedded in the Servier architecture. So this is Spartners, and Spartners is 2,000 square meter of independency, of innovation, and it hosts companies that are independent from Servier and that want to have labs, very full equipped labs. On top of that, services around their science to really accelerate their R&D. So this is not only an incubator, this is really an accelerator, but it's a physical building where we can find desk for, I would say, offices, and sure, but also full equipped labs. And that's where you would be hosted during the long period of this project, 3 to 5 years. You would have the chance to be part of this ecosystem there in this building. So in Spartners, where you could find entrepreneurs, you could find also all the services coming from BioLabs, which is our operator, because there is no collaborator from Servier in this little building. It's operated by BioLabs, the well-known and famous network of incubator, very famous in North America and coming in Europe with our incubator. So maybe the slide after. Yeah, so Spartners, as I said, it's there, and it's in front of your eyes. It's a real, true photography. So you have, as I said, you have offices, you have network and lounge space. You have very, very cutting edge technology in there. We have more than 2 million euros of equipment, scientific equipment, which is not trivial. And on top of that, the entrepreneurs then can also access to the brain of our collaborators in terms of mentorship, in terms of experimental advice or guidance. So this is a super environment for them to help. And I can tell you that all the collaborators coming from Servier play the game, and they are very willing to distribute their science to our resident. But for you, this is a bit special because you would be finally, it's another way to have a resident, but you will be so close to our teams that it will for sure enhance the communication in a day-to-day basis, which is really important when we are working in innovation and in that kind of project. So yes, this is like the paradise of the search, the researchers, because you have just to do the science and you have plenty of other services to help you in terms of, I don't know, waste management, calibration of the equipment, maintenance of the equipment. You don't care of that. You are just doing science. And the residents themselves, they say, here at Spartners, we can focus 90% of our time to science, where in other biopark and standard or classical biopark, it's less than 50, maybe 50%. So yes, here you can just do science and discuss with a survey team to yes, for the better and the best of the innovation. So yes, I think that's it. Yes, just to say that you can see the smile on the middle of the image, which is Servier, the survey site, and the other bubble are the other institute or high schools or university that are very close to us. This is a super ecosystem. You see the line, the yellow line, which is the future subway. So it will change our life because it will be very easy for you to have after work in Paris, just after your day. And there is two districts, we can say, the Polytechnic district, which is on the right of the image, and we are in the Quartier du Moulon district, which is close to Centrale Supélec, École Normale Supérieure. And when I say close, it's less than 200 meters. So it's very, very close. I think that's it for me. If you have any questions,
Thank you, Céline. I will take over with a few words on oncology before Clément described in more detail the challenge. So oncology is very important in Servier. We have grown internally and through multiple successful late stage and commercial acquisitions those last 10 years. And I just want to highlight two of them: the acquisition of Symphogen in Denmark in 2020. And Clément will talk about this center, which is our center of excellence in antibody. And then 1 year after, the acquisition of Aegis that brought us into the targeted therapy field and also allowed us to have our research and development center in US. So on the next slide is just to highlight that in oncology, we are committed to continue to innovate, and our activity are anchored in 3 pillars. First, the precision medicine, we develop the right drug for the right patient. On the right, we want to maximize the synergy between internal and external. So external is super important for us. And finally, in the middle, we have selected a specific area to focus, both in technology. So in technology, we do small molecule biologics, including multispecific, and ADC, antibody drug conjugate. We have also focusing on a number of scientific areas, and what is important today for the call is the T-cell engager, which is in the immuno-oncology space. And then the disease side, we are also maximizing on the knowledge that we are gaining in clinic, in the indication we are in: brain tumors, GI tract, and hematological malignancy with acute myeloid leukemia. And Clément, I give you the floor to describe the challenge, and thank you for the slide.
Thank you, Véronique. Thank you, Céline. So yeah, now let's move a bit more details into the call. So the call is called New AI Empowered Platform for Sterically Guided Design of Bispecific Antibodies. So just a quick slide to begin, just to put the words, and everyone is on the same page on antibody generators. So this is a monospecific antibody that you see here, the white chain protein. This is naturally occurring in humans and many other species. So this type of antibody, they bind antigens they bind the epitope site on an antigen to their paratope. The paratope is what gives the antibodies the specificity. So this is, as I said, a very big protein with 2 identical heavy chain and 2 identical light chain. So the natural antibodies, they bind the same epitope at 2 sites. These are bivalent antibodies. Now we will discuss more specifically bispecific antibodies in this call, which are artificial constructs. So it's one format of bispecific antibody, and the important thing that it has one site to bind one epitope from one antigen, and one site to bind a second epitope from a second antigen. So this is just one example, and there are many formats for bispecific and even multispecific format out there. So why are we interested in bispecific? This is because a T-cell engager are mainly bispecific or multispecific molecules, and they are a very potent modality in oncology nowadays. So how does this work? So we have a bispecific construct here. This is an example, which binds the CD3 on one side and a TAA, a tumor antigen, on the other side. So through this bispecific construct, we can force a connection between the T cells, the immune system, and the tumor cells. By forcing this connection, cross-linking the cells, we are producing an artificial immunological synapse, which induces the T cell activation, which will kill the tumor cells through the release of enzyme and perforin. They will also release those other inflammatory cytokines and start proliferating. So to sum up, this kind of bispecific can make all the T cells in the patient specific for the tumor. So these are very potent molecules. So bispecific engagers have been approved since 2014. The first one, the blinatumomab, actually in this format, was the CD3, CD19 engager type. So it's been approved in Europe, in US. And starting from 2021, we have seen more and more approval of bispecific engager in oncology, both in hematological malignancies, such as multiple myeloma and other B-cell malignancies, but also in solid tumor, such as uveal melanoma and small cell lung cancer. And more are coming with great results in clinical trial, and maybe will be approved in the coming months or years. In Servier, we are very interested in this type of molecules. And as was said by Céline and Véronique, Servier acquired to develop an expertise in antibodies, Synfogen back in 2020, which has become the Antibody Center of Excellence. So Synfogen is based in Ballerup in Denmark. And it's a state-of-the-art facility for drug discovery and development of biologics therapeutics. They are a very well integrated part of Servier R&D now, and they are serving as the antibody center of excellence. They have not only a facility, they have also a lot of great experts that support our value chain with key differentiated competence. As you can see, they have all the competence from the beginning of a research project on biologics up until the commercialization. And they have also a strong willingness overall in innovation and to reinforce AI tools to support biologics generation. And actually, 2 experts will join us in Saclay for the coaching during the BobCon, 2 experts from Synfogen. So now let's take a look a bit at the current discovery process that we have for bispecific antibodies. So let's say that we want to produce a bispecific targeting antigen 1 and antigen 2. So what we do is we do an in vivo immunization or a phage display, for example, in vitro display, to generate candidates, monospecific antibodies against each target. So antibodies 1 and 2. Then we do a first round of selection on this type of criteria. So we want to make sure they are very specific to the target. We are looking for specific affinities also and diversity of sequence, for example. But at this stage, we don't do a functional screening because it's not relevant for bispecific engager. So we do manage to select and to reduce the number of candidates. But actually, to test our bispecific, we want to test them functionally. It's important to select the preclinical candidate. But currently, we have no good way to predict the best combination between antibody 1 and 2. And it's not enough to have the affinity of this criteria to predict. So we need to test a maximum of combination of antibodies to discover the best bispecific antibody. So it means even if we reduce, for example, to 4 and 4, we have a magnitude of complexity of candidates here, and we go back to 16 candidates. So it's a lot of resource and time to then do the functional assay and select the candidates. So this is where the call comes in. So how do we improve this discovery process? So first, we believe that epitopes, so the way the place where the antibody binds the antigen is and the epitopes combination, so the way this bind and this bind is play an important role on the bispecific functionality. So the object of the call is to add another tool to our toolbox to predict the best epitope combination, to add this to the antibody selection before generating the bispecific antibodies, to have an accelerated discovery. So if we add this tool, this is what it would look like, our process with this tool. We will still do have to generate antibodies candidates through immunization or in vitro display. That will not change. We will still do a classical selection, but then we would have this new platform for the selection of the best epitopes for each antibodies. And so even if we cut, you see about half the selection of antibodies here, instead of 16, we would have only 4 antibodies to test. So that would be significant resource time and time gain for us. Alternatively, we could also use this platform to predict the epitope before the immunization or the display, so we can skew the candidates toward the epitope that we want. That would be maybe even better, but that would be another way to do it. Okay, so what we have in mind to build this model during the 3 years with BioMed X. We would do iteration of the predictive model. So we would start with a structure-based prediction of the best epitopes combination. We could imagine a scoring based on criteria such as the membrane proximity of the epitope, an important characteristic for T-cell engager, the synapse distance between the two epitopes, the steric hindrance, and for that, we could give already input to kickstart the model with antigen structure and antibody candidates epitope that we have already in-house at Servier. The second step would be to generate the candidates according to this score. We could outsource the production of all the candidates, but the functional screening would be done in-house by the team of BioMed X. And again, we are functional. We know how to generate this type of data, and we have already data to kickstart the model for several targets in-house. Then we would integrate this data and review the model up until we have a good algorithm, a good model tool that we can use to just, at the end, generalize the model, and we will give just the structure, the epitopes, and it will give us a rank of the best candidates to start the bispecific generation. That would be the idea. So of course, the starting point would be maybe a bit simpler, but I don't know if simple is the right term for that, but we would start with a fixed format of antibody, and maybe we can fix also one epitope and only a bispecific, but what we would like, if we can, is to extend the platform to more formats of bispecific antibody. For example, we would like also to extend to trispecific or even multispecific antibodies. And also, for now, we have discussed mainly T-cell engager, so it's transbinding modeling. So antigen are not on the same cells, and we are looking to have the best synapse, but we could extend also to bispecific ADC. And here it is cis-binding modeling and different problems with where we want to have better internalization. We could also look at ways to find the best binding sequence. For example, for T-cell engager, we often want to have the binding first on the tumor antigen and after on the T-cell. So if we can take that into account, that would be absolutely great. So that's it for the science part. Just to remind you for the application, our objective to develop this AI-driven platform to enable the rational design of bispecific antibodies by accounting for steric and structural parameters of dual target engagement. So the challenge will be to design a machine learning models that integrates all of the relevant data to have a prediction of epitope combination and dynamic modeling with the different format, etc., and to validate this model with experimental system. So really across not functional, but cross domain project. So I will not go too long into that because John already gave details, but we are looking for work group leader, 2 postdoctoral researcher, and 2 research assistant with backgrounds in machine learning, structure modeling, antibodies, biologics, immuno-oncology, or a combination of this background. But we are very much looking forward, as John said, as people open to collaboration, very open-minded, we like innovation, and also we are welcoming anyone from everywhere across the world, of course. So yeah, And of course, analytical and problem solve with kids, but you guessed it, I think it will be a tough challenge. So, so yes. And I'm done. Thank you.
Thank you very much for that excellent presentation.
Thank you.
So now we will move on to the Q&A portion of this webinar. So we already